Okay, so thank you for the introduction. I'm David Hathcock, a postdoc at IBM Research, and I'm going to tell you about work primarily done with Yuhai Tu and Chiwei Yu, who's a grad student at Princeton but has visited us for internships over the summer. And we've been working on understanding how dissipation enhances adaptation and sensing in bacterial chemotaxis. Um, so I thought I'd start with this old video from Howard Berg's lab showing the run and tumble swimming behavior of E. coli bacteria. And if you watch closely, you'll see that they swim roughly in straight lines and then occasionally tumble, flail around, and change direction and head off swimming in a new direction. And one of the reasons they do this is to perform chemotaxis by strategically choosing when to tumble. They can bias their motion up or down a gradient in chemical concentration, for example, to swim towards areas with more nutrients. And underlying this binary decision between running and tumbling is a simple biochemical signaling network. So here's roughly how it works. There's receptors on the cell surface that can bind to various extracellular ligands, and binding induces a conformational change that modifies the activity of a kinase key A, which is responsible for phosphorylating key Y, which is a messenger protein that carries this message to the cell motors and modulates this run and tumble swimming behavior. So this is a simple input-output signaling circuit that propagates the external signal to an intracellular carrier. And then on top of this, there's also an adaptation mechanism. There's enzymes KeyR and KeyB that can methylate and demethylate the receptors. And this shifts the downstream response across a wide range of concentrations, allowing the cell to maintain sensitivity to a variety of environments. So a little more detail on what that looks like. Here's a typical response curve for the KYP concentration as a function of the external ligand concentration, as a typical sigmoidal response. And if the cell starts off in this position A, near the sensitive regime of the response, and then comes upon a spike in ligand concentration, this induces more binding to the receptors, shuts off the kinase, and the KYP concentration drops following this response curve. Then on a slightly longer timescale, the adaptation kicks in. And this, as I said, shifts the response up in ligand concentration via the methylation of these receptors, returning the cell to the sensitive regime of the response. So with this adaptation, the cell maintains sensitivity to its current environment and is basically constantly resetting its scale for what's considered a large concentration. And this is really how it's able to move up gradients in chemical concentration because it's constantly resetting this scale and looking for even larger concentrations. Okay, so that's basically how the system works. And the other kind of key aspect that I want to understand in this talk is the spatially extended structure of this chemosensory system. So these receptors aren't isolated on the cell surface. They form this very sophisticated protein complex. The receptors, along with these other proteins, key A and also key W, which is kind of a coupling protein that is responsible for the formation of this lattice. So there's this extended cooperative structure, and by cooperative, I mean active receptors tend to propagate their activity to their immediate neighbors. So in this talk, we'll want to understand kind of how this interacts also with the energy dissipation that drives the signal propagation. This phosphorylation reaction is powered by burning ATP hydrolysis, burning energy to propagate the signal. Okay, so before I get into kind of what we've done on this topic, I want to zoom back briefly and tell you kind of traditionally how theoretical statistical physicists have built simple models to understand the response behaviors of the system, actually ignoring the two key properties I just mentioned, both the dissipation and the spatial structure. And kind of the key insight is that chemosensing is a cooperative allosteric process. As many of you probably know, allostery is this phenomenon where binding of a substrate to a site on an enzyme induces a long-range conformational change that influences the far-away active site of the enzyme. And this is exactly kind of what these chemoreceptors are doing. The binding induces a conformational change that propagates to the tip of the receptors and controls the activity of this kinase. Okay, so there's when we combine multiple binding sites or multiple receptors in this case together, there's kind of a classic old model for describing the cooperative allosteric um, uh, conformation changes of this sort of system known as the MWC model. So here we're going to consider a system of N receptors, and each receptor has a binary occupancy degree of freedom. Either it has a ligand bound or not bound. And then the key assumption in this model is that this cluster of N receptors acts collectively. They're either all active or all inactive. So there's just a single binary degree of freedom S that describes the cluster activity. So here I've written down kind of a statistical physics formulation of this MWC model where we have these binary encoded states, and they have different energy levels between these various states. And these energy levels are determined by a few parameters. There's a chemical potential-like term that depends on the external ligand concentration and the dissociation constant. This is basically the propensity to bind to the receptors. There's some energy difference between the active and inactive states of the cluster. And then there's a coupling term for how binding impacts this free energy balance between the active and inactive states. So this is a very simple statistical physics model. We can use Boltzmann statistics and partition functions to write down the average activity and average binding as a function of ligand concentration with these simple analytical forms. And I just want to give you one example kind of historically of how this has been used to nicely capture the response behaviors of the system. So this is a paper from Yuhai and Howard Berg, now 15 years ago. And this measurement I'm showing you is the kinase activity, or the QIP response, which is measured using FRET, a fluorescence imaging technique. And they measure this as a function of the external ligand concentration. And then the different colors here are different mutants of E. coli, where they've knocked out the adaptation enzymes and they fix the methylation level of the receptors. So as I told you before, methylation is this mechanism for adaptation or shifting the response curves across a range of ligand concentration. And so this measurement very explicitly shows how this shift happens as the methylation changes in the receptors. So to capture this kind of broad range of response data, all they had to do was make kind of one additional assumption for how the energy scales in this MWC model, if I go back quickly, depend on methylation, and they made just a linear scaling relation with methylation. And this gives you a model with, I think, 4 or 5 parameters that consistently fits this full range of response data. And you can extend this also to understand time-dependent responses and more complicated things as well. So this is just one example, but there's kind of a broad history in the early 2000s using MWC model and Ising type models as well to qualitatively or quantitatively understand a wide range of response behaviors of the system. But kind of what all of this work neglects is the energy dissipation that's involved in the signal propagation of this process. And so what I want to tell you about today is our work that has identified in kind of more recent experimental literature 2 signatures of the underlying dissipation that's involved in the signal propagation. And so I want to tell you kind of two brief stories about how we've identified these signatures of dissipation and then what they teach us about the underlying microscopic mechanisms for signal propagation in the chemosensory array. So I'll start out with this story about disproportionate adaptive shifts in activity and binding. Okay, so this was a measurement made in Jerry Hazelbauer's lab. And this top set of plots is showing basically an analogous measurement to what I showed you earlier. It's showing the QIP response as a function of the external ligand concentration. Again, this is the output of the signaling circuit as a function of the input concentration. And again, they've repeated this for mutants with fixed methylation, so you see the shift of the response across a wide range of concentrations. Now, they also measured the binding response curve. They measured the amount of bound ligand, again, as a function of the ambient or free ligand. And the interesting result they found is that as you change methylation, these curves shift much less than the downstream response. So here I've plotted them together, and you see there's a huge kind of 2 order of magnitude disproportionality between the adaptive shifts on the input side of the signaling circuit, these orange curves, and the output of the circuit, these blue curves. So the claim is that this is a fundamentally non-equilibrium behavior. I don't have time to kind of go into the full details of that, so I'll point you towards our paper for that. But I want to give you kind of an illustrative explanation using this MWC model. So as we've already seen, this MWC model, if we add methylation dependence to the, for example, the energy between the active and inactive states of the cluster, it nicely quantitatively captures the response behaviors of the system and the shifts with methylation across a wide range of concentrations. If we ask what the binding response, kind of the average sigma is as a function of ligand concentration, predicted by the MWC model, we find these solid and dashed curves for the 2 different methylation levels. And the different color here is just testing different cluster sizes in the model. And what you see is that these curves spread across a similarly wide range compared to the downstream output response, which is inconsistent with this kind of very narrow shift that's measured in the data. And the intuition behind what's going on here is because this model is equilibrium, there's kind of a reciprocal relationship between the downstream QIP response and the upstream binding response. So if we modify this energy via methylation by a dramatic amount that dramatically shifts this response across such a wide range, it feeds back through this interaction term to the binding response and shifts these curves across a similarly wide range. So that's kind of the intuition for why this model and also a broader class of equilibrium models can't capture this input-output relationship. So our resolution to this is kind of to make the simplest possible adaptation that adds in the dissipative phosphorylation, dephosphorylation reaction cycles that we know are underlying this signal propagation. So this cartoon is basically showing a schematic of our model. We're going to still have this sort of MWC interaction between ligand binding and the receptor conformations. But then the receptor conformation is controlling the on-off switching of this kinase, which is responsible for the phosphorylation of KY. So for simplicity, we'll just use a binary representation of the average phosphorylation state inside the cell. And when the receptors are on, they do this phosphorylation reaction. And then both when they're on or off, the dephosphorylation is catalyzed by a different enzyme in solution. So that happens regardless. But what this gives us is basically non-equilibrium driving cycles that are involved in the signal propagation. The next question is, how does methylation come into this model? And before we saw that if it modifies an energy in this MWC Hamiltonian, this propagates back to ligand binding and shifts the binding curves across a wide range inconsistent with the experiments. So our proposal is that methylation might modify the energy barrier to this phosphorylation reaction. And because the system is driven out of equilibrium, modifying this energy barrier impacts the non-equilibrium steady state of the system and can have a similar effect shifting the downstream average response curves of the system. So if we do some analysis and work out the average activity that comes out of this model, in the large dissipation limit, we actually find a functional form that's very similar to or actually exactly mappable onto the MWC model, but here it's coming from a very different mechanism. Rather than modifying one of these MWC equilibrium energies, the shift in the downstream response is coming from lowering of this reaction barrier for the phosphorylation reaction. So this gives us a mechanism that doesn't feed back to the binding response and allows us to simultaneously capture the broad adaptive shifts in the downstream of the signaling circuit and the narrow adaptive shifts on the input side. Okay, so that's kind of all I want to tell you about that. We can use our model also to study how things depend on the level of dissipation and interesting things like that. But for the purpose of time, I just want to leave you with this final thought for this part of the talk, which is that this model kind of gives a new hypothesis for the microscopic mechanisms underlying the uh, allosteric propagation of the signal within the receptors in the chemosensory array. So previously, it was thought that ligand binding basically um, impacted the free energy balance between these on and off kinase states, and that methylation was kind of undoing that conformational change or undoing the balance between those two conformations. Um, so in other words, both ligand and methylation were impacting the free energy balance between states in this of the same conformation within the receptors. We've proposed that methylation modifies the energy barrier to phosphorylation rather than the balance between the on-off kinase states. And one mechanism for this would be a distinct conformational change within the receptors that basically encodes this and propagates this to the kinase. So this is kind of a proposal we have, and there's some preliminary evidence in support of this. Single molecule FRET experiments on receptors in Gabriella Schlau-Cohen's lab at MIT that are really showing kind of distinct conformational effects due to ligand and methylation. Okay, take a sip of water quick. Now I want to tell you about the second story, which again kind of gives us new insight into the underlying dissipation of this dissipation underlying the signal propagation, which is this measurement of spontaneous switching and time reversal symmetry breaking. So this measurement was made in Tom Shimizu's lab a couple of years ago, and they found this surprising result that even in the absence of any external signals, there's no ligand in the system, the kinase activity spontaneously switches between fully active and fully inactive signaling states. So these time series I'm showing are single cell trajectories of the activity inside cell as a function of time. Again, without any external stimuli. So we have the spontaneous switching. This is already kind of an indication of the collective behavior of the system. These sort of switching and fluctuations that are observed can only really happen if you have an extended lattice of receptors that are collectively switching on and off in this way. From these dynamics, we can extract various timescales. There's dwell times, how long the system stays in the active or inactive state. And there's also transient switching times, how long it takes when the system actually executes a switch to go from active to inactive. So the dwell times are reasonably long. They're 50 to 100 seconds, and they're exponentially distributed. And the switching times have this kind of well, maybe not surprising, but interesting asymmetry between them. The switching times to the inactive state tend to be longer than the switching times to the active state. So this plot's showing the distribution of these times for two different types of receptors, and the switching to the on state, tau plus, are these two distributions, and they tend to be shorter than the switching to the off state. So there's this explicit breaking of time reversal symmetry in these transient switching times, and you can actually explicitly show for a wide range of systems that if you obey microscopic detailed balance, meaning your system's in equilibrium, there should be an exact symmetry between these two switching forward and backward switching times. Okay, so we wanted to develop kind of a minimal extended lattice model for this non-equilibrium extended lattice model for the system to understand these dynamics and also their implications for signal processing. So there's kind of three core components to the lattice, to the model. We have a lattice of many nodes, and each is driven out of equilibrium. So there's dissipation. So for simplicity, we've taken just a 3-state cycle, and there's dissipative driving that tends to push the system counterclockwise through the cycle. And this is roughly set up in analogy to the QI binding and phosphorylation cycle that's driven by the kinase in the system.
Just to let you know, about 5 minutes, just to.
Sure,
Yeah.
great. Yeah, the second key ingredient is cooperativity. So there's cooperative interactions between these nodes. If one is active, it tends to activate its neighbors. And then finally, how is the signal propagated to the lattice or encoded? As I suggested in the previous part of the talk, this is captured through an effective barrier shift to the phosphorylation reaction. And you can think of this kind of an analogy to like an icing model as being an external field that's applied to the system. So if we look at the dynamics of our model, it exhibits this very similar spontaneous switching between active and inactive states, at least if the coupling strength is sufficiently strong. If we apply this external field or effective barrier shift, we can bias the signal towards more active or less active signaling states. And then in particular, we see similar statistics for the switching times. The dwell times are long and exponentially distributed, and there's an asymmetry between the forward and backward switching times between these active and inactive states. We studied a little more systematically how these switching statistics depend on the two key parameters in our system, dissipation and coupling. And dissipation primarily affects this asymmetry between the switching times. So as I decrease dissipation, the forward and backward switching times converge to the same value. So the fact that there's a large asymmetry in the experimental data indicates that the system is highly out of equilibrium. The coupling primarily affects the dwell times, how long the system stays in the active or inactive state. And in analogy to maybe an icing model, if you increase the coupling in a finite size icing model, the two magnetized states, the effective barrier between the two magnetized states grows, and so their lifetimes grow exponentially. And while the system's out of equilibrium, kind of the same intuition holds. So we see an exponential growth in the dwell times as we increase the coupling. And this puts a reasonable constraint on how strong the coupling in this lattice can be, because if it were too strong, we would never observe these switching events on reasonable experimental timescales. So I'm not going to claim that the system's fine-tuned towards criticality, but the coupling has to be close enough to the critical point of the lattice, roughly within 20%, so that we observe these switching events on a reasonable timescale. So this leads us to the conclusion that E. coli is operating near this highly non-equilibrium, or at least reasonably close to a highly non-equilibrium critical point. Okay, with the final minute or two, I just want to give you some intuition for what implications this has for the signal processing behavior of the system. So what we found in our model is that there's a fundamental trade-off between the speed and sensitivity of the response. So here I'm plotting the sensitivity, how sharp the response curves are, and the response speed against each other. And each of these colors is a fixed dissipation. So as we tune the coupling strength in the lattice, there's a trade-off between speed and sensitivity. This is, I think, somewhat intuitive. If you increase the coupling, the lattice tends to respond together, so that gives you sharper, more sensitive responses. But as I showed earlier, this increases the kind of dwell times in the system, the characteristic relaxation timescales, which gives rise to slower responses. So there's this fundamental trade-off between speed and sensitivity, and one role of dissipation in the system is easing this trade-off. It pushes these curves towards the upper right, allowing the cell to obtain both a faster and more sensitive response. And then finally, I just want to also mention that this can lead to a more robust signaling behavior as well. So suppose there's some operational thresholds in both speed and sensitivity that are required to effectively sense and respond to the environment for the cells to survive. This means that there's then a minimum dissipation level that's required to basically meet both of these thresholds and successfully sense the environment. But if we operate exactly at this minimum level, this requires fine-tuning of the coupling strength or other parameters in the system to this point where this cell can respond both fast and sensitive enough. Increasing the dissipation above this threshold gives rise to a wider range of couplings for which the cell can obtain both fast and sensitive response. Okay. Okay, so I think I'm just about out of time. So I'll leave you with kind of this conclusion. We've identified some signatures of non-equilibrium dynamics in more recent experimental probes of this chemosensory system in E. coli. And we've shown how energy dissipation enhances the speed, sensitivity, and adaptation in these systems. If you're interested in more details, please check out our papers from the last couple of years. Thanks.